Welcome to the webinar, Report Back from SGO, What's the Latest in Ovarian Cancer? I'm Maggie Nicholas-Alexander, the Ovarian Cancer Program Director at SHARE. Before the presentation begins, I'd like to tell you a little bit about SHARE. SHARE is a nonprofit organization that has been helping people through breast, ovarian, and uterine cancer for the last 45 years. By offering the support of those who have been there. SHARE offers many services, including a helpline, support groups, and educational programs. All of our services are free of charge to participants. For more information, visit our website at sharecancersupport.org. All participants will be muted during the presentation. When Dr. Liu finishes presenting, we will begin the Q&A discussion. You are welcome to submit questions during the presentation through the Q&A section at the bottom of your screen. The chat section will be disabled. When asking questions, remember that the presenter is unable to give specific medical advice. So please keep your questions general in nature. This webinar is being recorded and it'll be available on the SHARE website soon. Now, I'd like to introduce today's speaker, who we are so excited to have here. Dr. Liu is an assistant professor of medicine at Dana-Farber Cancer Institute and Harvard Medical School. She is a medical oncologist specializing in the treatment of women with gynecologic malignancies and is the Associate Chief and Director of Clinical Research for the Division of Gynecologic Oncology at Dana-Farber Cancer Institute. Dr. Liu's research focuses on identifying and validating novel therapies and therapeutic combination for gynecologic cancers. We're so excited to have you here, Dr. Liu, and I'll turn it over to you now.
Thank you so much. And thank you for that introduction and for the invitation to speak today. I'm very excited to be able to bring this report back from SGO about the most recent developments in ovarian cancer therapy that have been shared from the meeting. I'm going to share my slides. All right. So this is the report back from SGO. What's the latest in ovarian cancer? And just to take a step back, oh, these are my disclosures, apologies. And then just to take a step back and think about where we are in ovarian cancer in 2021. And, in some ways, things are very similar to where we've been for many years. Ovarian cancer remains the 5th leading cause of cancer death for women in the United States, but things are also changing. There are about 21,500 new cases annually and a little bit under 14,000 deaths annually in the United States. And that's actually a change. So year by year, over the past decade or more, we have seen both the number of new cases and, importantly, the number of women who die of ovarian cancer decreasing in the United States. So we are making progress. When we think about it sort of, from, how do we think about treating ovarian cancer as a whole, taking that step back again, when women are initially diagnosed with ovarian cancer, the initial standard of care is surgery and chemotherapy. And unfortunately, there are a number of women where the cancer comes back. And in that first recurrence, depending upon the timing and where the cancer comes back, we think again potentially about surgery, we think about chemotherapy. And once ovarian cancer comes back, unfortunately, it is a cancer that becomes more chronic. It is more like a chronic disease where it can go into remission for certain periods of time, but then does tend to come back again And those recurrences, subsequent recurrences, are typically treated with chemotherapy or some sort of systemic therapy, and continuing on. And the questions that we've asked ourselves for many, many years are, well, how do we make our ovarian cancer treatments more effective? If this is our paradigm for where we are right now for a woman who has been diagnosed with ovarian cancer, how do we make our treatments better? How do we help women live longer? How do we move maybe even towards a cure? Can we improve our initial treatments in ways that will prevent or potentially delay the cancer from coming back? So really changing sort of this arrow here. One of the big questions I think that has been asked for many years is, can immunotherapy improve treatments for women with ovarian cancer? Is this a modality that we can turn to really change the curves and the trajectories that we've seen over these years? And finally, I think the last question that I often think about is, what are the novel therapeutic targets? What are new ways of attacking ovarian cancer that we haven't done before that can provide new treatment options for women whose cancer have come back and sort of are no longer responding to standard chemotherapies? And if those are effective, are those things that can eventually sort of move into these earlier treatment stages? One of the things that we've really thought about over the years is this idea of improving initial treatment. And many of you may have heard of this idea called maintenance therapy, which is that, if we look at that initial paradigm and we say, okay, we treat with surgery and we treat with chemotherapy, and that for some number of women after some period of time, the cancer comes back. The question that we ask is, if we add additional therapy after you do surgery and chemotherapy, can you prevent or delay disease recurrence? So can you go on some sort of maintenance type of therapy and either keep the cancer from coming back for a longer period of time, or in the best case scenario, keep it from coming back at all? And this is a question that has been asked for many years. One of the first questions of maintenance therapy was with a drug called bevacizumab. This is an antibody that blocks a receptor called VEGF, or actually it doesn't block a receptor called VEGFR, but it blocks a signaling pathway called the VEGF receptor signaling pathway. And that's a pathway that causes the growth of blood vessels. Now you can imagine cancers actually really are dependent upon that pathway, because cancer cells are growing abnormally and they need to recruit blood vessels so that they have oxygen and nutrients and all of the things that cells need to grow. And many years ago, it's been shown that if you take a drug like bevacizumab and you use it in ovarian cancer, that it can have activity. And about 10 years ago, this question was asked in 2 large randomized phase 3 trials, which was to say, if we added bevacizumab to chemotherapy, could we make a change? Could we make our therapy better? These are what are called Kaplan-Meier curves. You'll see these throughout this presentation. Kaplan-Meier curves are sort of a visual representation of the time that it might take before cancer comes back across a number of women. And what you can see here is this sort of is women whose cancer has not come back. And you can see sort of as time goes by, a number of women, their cancer comes back. And when you add bevacizumab to chemotherapy, you can see this in the red line here versus the blue line here, that time extends and extends sort of on average somewhere between 2 to 4 months. And this one is not a colored graph, but again, this darkest line compared to this very light line is with bevacizumab and without bevacizumab. And again, you can see that bevacizumab is in fact effective at extending the time before cancer comes back when it's given together with chemotherapy initially and then continued on as maintenance therapy. But it turned out that when we looked at the overall time that women lived in these 2 trials, the bevacizumab maintenance could help women's cancer stay in remission for a longer period of time initially, but it ultimately didn't extend how long women lived. That being said, being cancer-free for a longer period of time is very valuable. And so the FDA did approve bevacizumab as maintenance therapy for newly diagnosed ovarian cancer a few years ago. And this is one of the standards of care that we sometimes consider. The newest kid on the block, if you will, is the PARP inhibitors. And the PARP inhibitors now have been sort of, many of you may have heard of them or have heard them discussed over the past 10 years or so. These have been one of the most exciting advances in ovarian cancer in this period of time. And the trials have started to look at, can you give PARP inhibitors as maintenance therapy? PARP inhibitors are an ideal type of drug for maintenance therapy. They're oral pills. They are generally, pretty well tolerated. They do have side effects, but the side effects are much less than doing chemotherapy. They allow people to live their usual lives and continue to work, to travel, to do all sorts of things and not feel tethered to their oncologist care. PARP inhibitors work with this idea that they block the repair of DNA. And DNA has a number of ways within a cell, a normal cell, that it's repaired, but there are two major pathways. One is something that is called base excision repair. That's one where PARP is a very active player. And then there's another type of DNA repair called homologous recombination. And that's where a number of genes, including the genes BRCA1 and BRCA2, are very important players. In normal cells, both of these are active and intact pathways, so that if you have DNA damage, the cell can repair its DNA depending upon the type of DNA damage by either of these pathways and does not get into any trouble. In cells that, for example, have a BRCA mutation or another alteration that affects homologous recombination, that happens about 50% of the time in high-grade serous ovarian cancers, which are the most common type of ovarian cancer, the cells still have intact base excision repair. And so it turns out that they can still repair their DNA damage through this base excision repair pathway, and they don't really get into trouble. However, and then if you take normal cells that don't have any loss of homologous recombination and you block them with a PARP inhibitor, they lose the base excision repair pathway, but the homologous recombination pathway is intact. They still can repair their DNA well. They don't get into any trouble. But now if you have a cell, and as I mentioned, about 50% of ovarian cancer cells have loss of homologous recombination, a trait that we call homologous recombination deficiency, or HRD. And then you block PARP in these cells, they lose this base excision repair pathway. Now these cells lose two of their most effective ways of repairing DNA. They don't repair their DNA, and this ultimately leads to cell death. And this particular set of interactions was described about 15 years ago in paired papers that came out from the laboratory setting in 2005. And we've been developing PARP inhibitors in ovarian cancer since then. They have shown effect in a number of settings, and starting about 2 years ago, we had 4 trials of PARP inhibitor maintenance therapy for women with newly diagnosed ovarian cancer. And these have been one of the most exciting developments that we've really seen. These 4 trials, you may see their names: SOLO-1, PRIMA, PAOLA-1, and VELIA. And again, not to say that you're to start interpreting these Kaplan-Meier curves so that to understand things, but what I'm going to show you is that, again, across all 4 of these trials, SOLO-1 is women who had BRCA-mutated ovarian cancers. You can see this vast change in the curves. This red curve with olaparib maintenance therapy, so with a PARP inhibitor maintenance therapy compared to a placebo, a sugar pill, you can see this really prolonged extension in how long it was before cancer came back, including in a number of women where the cancer has not shown evidence of coming back. Again, in PRIMA, an extension of these curves. This is in all women, so both women with or without BRCA-mutated ovarian cancers. PAOLA-1, again, in women with or without BRCA-mutated ovarian cancers, but now in combination with bevacizumab. And then finally, Velia, which actually took olaparib, a different PARP inhibitor, and put it in combination with chemotherapy. Again, when women with and without BRCA mutations. And in every single one of these trials, we can see that PARP inhibitors significantly delay the time to ovarian cancer recurrence. The activity is highest in women with a BRCA1 or a BRCA2 mutation in their tumor. So you can see that the difference in the SOLO1 curve is the most prominent. And we see this, I haven't shown you the subsets in here, but this is seen also in the PRIMA, PALA-1, and Velia trials. But in both PRIMA and Velia, there was evidence of benefit across all women, albeit to a different extent. So this is sort of where things have sat. And I just wanted to provide that context to sort of then talk about these updates that we have from SGO that occurred in March of this year. I think one of the most exciting updates is, in fact, an update to one of these trials that I've talked about, the SOLO1 trial. And this is of maintenance olaparib. Olaparib is one of the PARP inhibitors in women with a BRCA-mutated ovarian cancer. And now looking at women now 5 years out from having gone on the SOLO1 trial. So when SOLO1 was first presented 3 years ago, we did not have this amount of follow-up time. So we didn't know what was going on sort of afterwards. The way SOLO1 was designed, this trial was of women who had a newly diagnosed advanced ovarian cancer. They had to have had a BRCA mutation. They had to have shown a response to chemotherapy, and that happens the vast majority of the time. Women were randomized to receive either olaparib for 2 years, so just 2 years of treatment, not indefinitely, not forever, or to get placebo for 2 years, so a sugar pill. And here is the Kaplan-Meier curve, now 5 years of follow-up from the SOLO1 trial. And what you can see here is that 2-year treatment cap. This is when women ended their olaparib maintenance therapy, and yet we see continued benefit. This is incredibly exciting. It says that we can treat with 2 years of drug, but the benefit of having done those 2 years of maintenance therapy extends. You don't have to continue on PARP inhibitors for 2 years, for an indefinite period of time. And so that's really exciting. I think what's also very exciting to me is, again, looking at these curves 5 years out, we're seeing that almost half of these women now, their cancer has not yet come back. And that really raises my hopes that not only are we potentially delaying the cancer from coming back, we might be preventing it. That for some of these women, by going on a maintenance PARP inhibitor, we are indeed preventing cancer recurrence. So this is a very exciting set of data, the update from the SOLO1 trial that came from SGO this year. I think I've talked through many of these points. These are my takeaways, is that there is this benefit that remains at a 5-year follow-up, even after just 2 years of olaparib maintenance therapy. It makes it incredibly important that we are testing for BRCA mutations in newly diagnosed ovarian cancer. One thing we don't yet know, but I think these data sort of give us high hopes for, is that we don't know if we're helping women live longer by doing these 2 years of maintenance therapy, but the change in these curves is really impressive. I think that it really, suggests that we might be doing that. We have to wait for the data to mature, but this is a real possibility. One thing we don't quite know yet is PARP inhibitor maintenance for women who don't have a BRCA mutation. So the trials that examine this, the PRIMA, the PALA-1, and the Velia trials, those trials were a little bit later in their development compared to SOLO1. So we don't have that same 5-year follow-up yet for those trials. And those are the trials that enrolled women who did not have a BRCA mutation. But that information, I think, will become known to us in the next few years and will, again, be very important and hopefully very exciting for us to see. Moving on from the update from SOLO-1, I think one of the other very important studies that came out and was updated at SGO was the question of adding immunotherapy to chemotherapy and bevacizumab in newly diagnosed ovarian cancer. This is the IMagyN50 trial. This is a trial that has been going on, and originally was presented at ESMO in 2020, so about 6 months ago. And we've been waiting to understand sort of whether these additional results could be of, could give us additional information about when immunotherapy might be something to do. Just a little bit of background. We've been trying to add immunotherapy to chemotherapy in ovarian cancers. We've been very inspired by the success in other types of cancers and immunotherapy for a number of reasons. If you look at ovarian cancers under the microscope and think about the activity of immune cells in ovarian cancer, this is one of the cancers where it feels like taking the brakes off of the immune system and getting it more excited should make a difference. There have been really nice preclinical, so lab-based models that have suggested that putting immunotherapy together with chemotherapy in ovarian cancer, as well as in other types of cancers, is a way to make immunotherapy better. But it hasn't really borne out for us in clinical practice so far. The Javelin Ovarian 100 trials and the Javelin Ovarian 200 trials are trials of chemotherapy plus an immunotherapy drug called avelumab that were reported out not at this year's SGO meeting, but last year's SGO meeting and the SGO meeting in 2019. So 2 years worth of SGO meetings looking at this. And you can see, again, these Kaplan-Meier curves in this particular case. This is chemotherapy. In the yellow is chemotherapy plus, the yellow and blue is chemotherapy plus immunotherapy. And the red here is chemotherapy alone. And unlike what we see with the PARP inhibitors and with bevacizumab, the immunotherapy is not adding to chemotherapy in this trial, which is a newly diagnosed ovarian cancer, or in this trial here between chemotherapy in green and chemotherapy plus immunotherapy in blue in recurrent ovarian cancer. And so these are data that we've had before. I think many of us were very hopeful and excited about the IMagyN50 trial because it added bevacizumab. And there have been, again, good preclinical data that suggest that bevacizumab and sort of all of the pathways that bevacizumab inhibits will make the immune system better at fighting ovarian cancer in and of itself, and then adding in immunotherapy would potentially help that even more. And so IMagyN50 was again designed as a randomized trial. Women with newly diagnosed ovarian cancer who had never been previously treated were randomized to get either chemotherapy with bevacizumab plus a placebo, and then continued on bevacizumab plus a placebo, or to get chemotherapy and bevacizumab plus a immunotherapy called atezolizumab, and to continue on that afterwards after completing chemotherapy. And unfortunately, these were the results that were seen in the IMagyN50 trial. You can see here that the curves are not separating, so immunotherapy did not add to the activity of chemotherapy in this trial. In women who had this marker called PD-L1, which we think in other cancers we have seen can sometimes be a biomarker of somebody who is more likely to respond to immunotherapy. Maybe this curve separated out a little bit, but it was not very significant. And so again, in this trial, adding immunotherapy in this way didn't seem to give benefit. And the update from SGO was that even if you looked at different biomarkers, including BRCA status, there was not a difference by adding immunotherapy to chemotherapy. This is not to say that we shouldn't keep looking at immunotherapy in ovarian cancer. The drugs that are currently approved by the FDA, these PD-1 inhibitors and PD-L1 inhibitors, are one way to make the immune system more active, but there are actually a whole host of signaling factors that help to modulate the immune system. And so I think there are still many, many things that we can do to try and activate the immune system in ovarian cancer. But we have to be thoughtful about it a little bit, to go back to the drawing board and think about what's the best way, what are the novel ways that we can think of applying immunotherapy in ovarian cancer. And I think there is a lot of research going on in that. Moving on from that and coming back to PARP inhibitors, and not as maintenance therapy, but the idea of, can you use PARP inhibitors as a treatment? How do PARP inhibitors compare to chemotherapy in women with recurrent ovarian cancer? And one of the studies that was presented at SGO was the ARIO-4 study, which really looked at the PARP inhibitor rucaparib and compared it to chemotherapy in women who had relapsed or recurrent ovarian cancer that had a BRCA mutation. And so this was a comparison of PARP inhibitors to chemotherapy. The design of this trial was women with recurrent ovarian cancer had to have a BRCA mutation. They had to have had at least 2 prior chemotherapies and never had received a prior PARP inhibitor. And these women were randomized to receive either rucaparib or to get standard chemotherapy that could be either paclitaxel, also called Taxol, if they were within 12 months of their last platinum-based chemotherapy, or platinum-based chemotherapy if it had been more than 12 months after the last platinum therapy. Here you can see the Kaplan-Meier curves in this particular case. So you can see that the PARP inhibitor seemed potentially just as good as chemotherapy, maybe even better, depending upon the setting, where rucaparib, in terms of that extension of the median of the progression-free survival. So again, this is that line of sort of how the time frame before the cancer comes back, that is extended sort of by having rucaparib instead of chemotherapy. I think that this trial really confirms what we've known, that PARP inhibitors are active in BRCA-mutated ovarian cancer as a treatment. That's certainly true for women who have never received a PARP inhibitor. In today's day and age, I would say to you that I think that every woman who has a BRCA mutation in their ovarian cancer should get a PARP inhibitor as maintenance therapy when they are first diagnosed. And so we don't know what will happen if somebody's had a PARP inhibitor before, but if somebody hasn't had a PARP inhibitor previously and has a BRCA mutated cancer, I think that we absolutely need to think about PARP inhibitors as treatment. Rucaparib had better activity than chemotherapy. There are some caveats to this. There are some sort of nuances to this study in terms of the choice of chemotherapy and who got what chemotherapy that mean that we have to be a little bit cautious in sort of interpreting and saying that anybody should get, a PARP inhibitor instead of chemotherapy. But for somebody who has a BRCA mutated ovarian cancer, who hasn't yet received a PARP inhibitor, absolutely, I would say that what ARIO-4 tells us is that PARP inhibitors by themselves can be considered as a real option for care, not just as maintenance therapy, but as a treatment for active ovarian cancer. How about PARP inhibitors as maintenance therapy? We talked about in the first line setting that the data from the trials that had women who did not have a BRCA mutation had not yet matured. The NOVA analysis did mature and was presented at SGO. This is data I think many of us have been waiting for and hoping to see. NOVA is a phase 3 trial that looked at maintenance PARP inhibitor with niraparib in women with recurrent ovarian cancer that had responded to platinum. In the original NOVA trial, women who had recurrent epithelial ovarian cancer who had responded to platinum were randomized. There were 2 groups, those with a BRCA mutation and those who did not have a BRCA mutation. They were randomized to then get either niraparib as maintenance therapy, so finish chemotherapy and then taking niraparib afterwards, versus placebo. And the original trial results, which were reported back in 2016, so 5 years ago, showed that in both groups, women with a BRCA mutation and those without a BRCA mutation, that there was significant benefit in delaying the growth or delaying the recurrence of ovarian cancer when niraparib was given as a maintenance therapy. The question that was asked here in the final analysis of NOVA was, did that change translate into women living longer? There are some issues about the NOVA final analysis, and I will say that this is complicated statistician type language that I myself look at and I'm like, okay, this tells me that we have to interpret these very cautiously. But basically what happened was that when they collected the data for these trial, there was some missing data. So after women came off of the trial and went on to subsequent treatments, not all of that data were collected. And what that means is that some of the data are incomplete. And so we have to be a little bit careful that this is not absolute truth. Some of it is projected. But what was seen and what was reported by Dr. Bachelonis at SGO was that when they do look at this in women who do not have a BRCA mutation, it is not clear that going on a PARP inhibitor as maintenance therapy is changing how long women live. So the benefit people are getting is, at least from what we can tell right now, predominantly in keeping the cancer at bay for a longer period of time. So being able to delay going on any further chemotherapy and taking the PARP inhibitor pills instead. In women with a BRCA mutation, it does look like there's a trend towards increasing, so helping women live longer. You can see from the orange curve to the green curve, there is that difference. And that's completely in line with, that's completely in line with what was presented at the ASCO meeting last year from the SOLO2 trial, which looked at olaparib maintenance therapy in women with BRCA-mutated ovarian cancers and showed that if you looked at the overall survival, so whether or not olaparib maintenance was helping women live longer, that in fact it looked like there was a trend towards a 13-month improvement in helping women live longer. And if you then adjusted for whether or not women got more PARP inhibitors, actually that improvement in sort of how long somebody lived was actually even substantially more. These results, I would say, really support that PARP inhibitor maintenance is important. I think I've talked about this certainly in the frontline setting, but for women who have a BRCA mutated ovarian cancer and who haven't had a PARP inhibitor before, if you haven't had it when you were initially diagnosed, then in subsequent diagnoses, I think it is really important that women with a BRCA mutated ovarian cancer get a PARP inhibitor. And finally, I think one of the other things that we've been thinking about are, what are the new therapies on the horizon? What more can we do? We've talked about PARP inhibitors, we've talked about immunotherapies. Are we developing other therapies? And this is this was in our first plenary session at SGO this year, which was talking about a really novel drug called AVB500 that targets a signaling pathway called Gas6-AXL. And this is a pathway that nobody really has ever attacked before in ovarian cancer. Gas6-AXL is this sort of signaling pathway here. AXL is a receptor tyrosine kinase. What that means is it sits in between, this is the cell membrane, so it sits on the surface of the cell, and it takes signals that are circulating in the bloodstream by this molecule called Gas6. And when Gas6 sort of hits and binds to AXL, a whole host of things happen which activate these downstream pathways, basically like signaling pathways. It's almost like, turning on lights in a row, or a domino effect that goes down into the cell and ultimately results in cell proliferation, so cells growing more, and cells survival, meaning cells that don't die when they're supposed to. It's an important pathway that's been identified in ovarian cancer as a pathway of interest. And it's been very hard to target because it turns out that, if you try and target with these small molecules like we've done for some other pathways, it's not very specific. You end up hitting a lot of things. AVB500 is actually this novel drug. What it is it's basically an antibody type drug that really binds very strongly to Gas6. And Gas6, as I mentioned, is the signaling molecule that then binds to AXL and causes that downstream sort of light up of these pathways that leads to cell proliferation and cell survival. And so what this AVB500 does is it basically soaks up all the Gas6 that's floating around in the bloodstream and keeps it from being able to bind to AXL and therefore keeps signaling from happening. At SGO were presented for the first time the results of the phase 1b trial that looked at combining AVB500 with chemotherapy. One of the things that we see is that Gas6 and AXL signaling seems to be one of the methods by which cancer cells can become more resistant to chemotherapy. And so AVB500 was combined with chemotherapy with this idea you could reverse the resistance. We combined it with two different chemotherapies. One was paclitaxel or Taxol, and the other one was pegylated liposomal doxorubicin, which many of you may have heard of as Doxil. This is what we call a swimmer's plot, which is to say sort of, you start at time 0, and this is the time that people have remained on study. And you can see that there have been a number of women on study who continue on study, who had a lot of benefit from this combination, mostly in combination with paclitaxel. And so this has been a really exciting sort of possible new direction and a possible new combination that we can develop in ovarian cancer to attack the cancer in new ways and try and keep it from becoming as resistant to our chemotherapies. This has led to the development of a phase 3 trial, which actually has just opened. And I think the first patient went on about 2 weeks ago, of weekly paclitaxel plus AVB500 in women with recurrent platinum-resistant ovarian cancer. This is going to be a phase 3 trial that's enrolling in the United States as well as internationally in Europe. It's led by Dr. Catherine Fu, who presented the results at SGO and actually has been critical in developing this particular drug all the way from the laboratory to the clinic. So it's a very exciting line of development. And in this trial, women will get either weekly Taxol, weekly paclitaxel with AVB500, or weekly paclitaxel alone, and will be followed throughout. And so this is a trial I think we're all very excited about in the ovarian community, and excited to see the first results of the activity of AVB500 plus weekly paclitaxel at the SGO meeting. So just to summarize, what I think were the take homes from our SGO meeting this year, the big things that came out of it in ovarian cancer. I think that more now, just as ever, we've really seen further evidence that PARP inhibitors are a very active and important part of our armamentarium against ovarian cancers. These are drugs that are most active in women with BRCA-mutated ovarian cancer. We now know and we have demonstrated that there's prolonged benefit for women in newly diagnosed disease who have a BRCA mutation, even after the PARP inhibitor therapy has been stopped. We have seen that there's activity in women with recurrent disease. We have seen that, we have to do more work to really understand how we effectively leverage immunotherapy in ovarian cancer. The things that we've done so far haven't moved immunotherapy forward in the field, but there's still much more to come. And so it's not to say it's the end of the story for immunotherapy in ovarian cancer, but that there are more directions that we have to continue to look at. And that novel therapies such as targeting of Gas6 and AXL continue to be developed and are exciting new directions where things are going. So I thank everybody sort of for the attention, the time, the invitation to speak, and I hope I've left us enough time to answer questions and have good discussion about it. Thank you.
Thank you, Dr. Liu. That was a great summary of the research that came out of SGO. And we definitely have a good amount of time for Q&A, so that's great. So let's start the Q&A. You can still submit questions in the Q&A section at the bottom of your screen. We will try to get through all of the submitted questions, but we may not be able to due to time constraints. And again, Dr. Liu cannot provide any medical advice, so please try to keep your questions general in nature. All right, so let's get started with some of the questions that were submitted. So unsurprisingly, we got a lot of questions about PARP inhibitors. And one question that I think is kind of interesting and comes up a lot is around the cost of PARP inhibitors. And, many women in the share ovarian community talk about the high cost of PARP inhibitors and, may depend on financial assistance to afford them. So do you think as these medications grow in popularity and continue to be used more, that there's any indication that the costs may start to drop?
So I think the hope, of course, is that the costs will start to drop. The PARP inhibitors, the pricing of the PARP inhibitors that has been set by the various companies that have developed them has been very high because they are monthly oral treatments. They are covered under prescription drug plans as opposed to sort of traditionally our infused therapies. And I think this has been a real gap that we've seen in the US healthcare system, that, as much as we push towards developing oral therapies for convenience and, for reasons that, you don't need an IV infusion, that has led to really a lot of financial toxicity because of the way the US healthcare system is structured and the way that oral medications are covered compared to IV infusion coverage through insurance. I can't speak to policy and sort of what will happen in terms of the cost of PARP inhibitors. Certainly, I hope very much that the costs will become more affordable. Each of the companies that makes the PARP inhibitors does have drug assistance programs, and that has been the predominant method that we have had to sort of rely upon to get coverage for PARP inhibitors for women who have very high copays or who have very high co-insurances for their PARP inhibitor coverage, depending upon their insurance plans.
Thank you. And I think it's a good reminder about those assistance programs that are available through the pharmaceutical companies. So thank you for that. And so another question about PARP inhibitors, but I think another important thing that people often wonder about, but could you speak to the difference between the three approved PARP inhibitors? So that's olaparib, niraparib, and rucaparib, and sort of like the difference between them and how they're used.
Yeah. So, I think that I will say that between niraparib, olaparib, and rucaparib, so there are differences in how they have been approved by the FDA. So there are differences in their approval in the first line setting. There are not differences in their approvals in the recurrent maintenance therapy setting. Those are sort of uniform. There are differences in their approvals in terms of treatment for recurrent ovarian cancer. So not maintenance, but therapy for active ovarian cancer. The approvals are really based upon how the trials were designed and how the trials supported them. I would say first off is that, in my personal opinion and based upon sort of what we know about the data from the trials, I don't think we've ever seen clear evidence that any one of these PARP inhibitors is more active or less active than the other two in any clinical setting. So I don't think about these as necessarily differentially active PARP inhibitors. We do know that they have different side effects, and we do know that there are different side effect profiles for them. So Niraparib is taken once a day, as opposed to rucaparib and olaparib. And so it is, for some people, especially people who pills are a lot, that, a once a day dosing can be more convenient. Niraparib does have a bigger effect on platelet counts. So it can cause platelet counts to drop more aggressively in the 3rd and 4th week or so after starting niraparib. There is a very specific dosing of niraparib based upon platelets and weight that has been shown to somewhat ameliorate that. But anybody I start on niraparib, I make sure that I am checking their blood counts every week for at least the first 4 weeks and typically the first 8 weeks after I start therapy, to make sure that there's not an issue with the platelet counts. And so that's a difference in terms of sort of quality of life and lifestyle, depending upon how easy it is to get blood work drawn. Niraparib can have some other effects, including causing some hypertension that we don't see with rucaparib and olaparib. Olaparib, as I've mentioned, is a twice daily dosing. It is probably a little bit more likely to cause anemia than niraparib, and so we do see that it can result in slightly lower red blood cell counts. Rucaparib has some particular chemical sort of qualities where it can cause your liver function test to get higher or to get the kidney function test to get higher. These are not actually things that turn out to be medically important. They're more things that we observe on the blood work. But they are also, again, a consideration when thinking about which PARP inhibitor to use. I think, again, the indications, rucaparib, niraparib, and olaparib are all approved for maintenance therapy in the recurrent setting. Olaparib is approved for maintenance therapy in the first-line setting for women with a BRCA mutation, and together with bevacizumab in the first-line setting for women who have either a BRCA mutation or who don't have a BRCA mutation, but have a tumor that is considered HR deficient by a test that is done through Myriad. Niraparib is approved in the first-line setting as maintenance therapy across all women, regardless of whether or not you have a BRCA mutation, but not in combination with bevacizumab. And rucaparib is not approved in the first-line setting. So there are differences in the approvals, but not necessarily because of activity.
Okay, yeah, thank you. That's really helpful because I know there's a lot of confusion around that. And so I think that's a really helpful summary. So this question goes back to immunotherapy. And there seems to be more use of the PD-1 L1 inhibitor, pembrolizumab or Keytruda in certain recurrent settings. Can you talk about that?
So there's a study called Keynote 100 that looked at pembrolizumab in women with recurrent ovarian cancer. The overall activity was relatively low. And so, I wouldn't say that there's a lot of data to say that we should be using PD-1 inhibitors or PD-L1 inhibitors by themselves in recurrent ovarian cancer. There are some women who really benefit. They have, marked responses, they last for a long time, and they clearly benefit from immunotherapy. The problem is we don't know yet how we identify those women. And that's, I think, that's why I was saying, it's not that I think we should give up on immunotherapy in ovarian cancer, but I think we have to be very thoughtful, and we have to go back to the drawing board to understand why immunotherapy works when it does, and how we make immunotherapy work in other ways when it isn't working already.
Mm-hmm. That makes sense. Thank you for that. And so going back to PARP inhibitors, because there's been a few variations of this question, and it's really just around how long patients can be on them, and what is the considerations around that in terms of whether they're on it 2 years or 3 years or beyond.
Yeah, so in the studies done in women with newly diagnosed ovarian cancer, The olaparib studies had women continue on olaparib maintenance for 2 years. The niraparib study, the PRIMA study, had the women continue on niraparib maintenance for 3 years. In both of those studies, the PARP inhibitor maintenance was stopped at that time, unless somebody still had really clear evidence of sort of measurable or active disease, in which case a small number of women did continue on PARP inhibitors indefinitely. I think that, there's been a lot of question, how long should you continue on PARP inhibitors? Do you need to continue them, indefinitely? Could you stop at 2 years? Can you stop at 3 years? I think that that's one of the reasons the update from SGO this year, that 5-year follow-up from SOLO-1, is so exciting, because it really suggests that you don't need to keep going, that 2 years of maintenance olaparib from the SOLO-1 study is enough, and that you're not getting more bang for your buck by continuing on PARP inhibitors longer. One thing we don't really understand in the, when women have PARP inhibitor maintenance in their cancers that have come back, they do continue on PARP inhibitors. All the studies continued on PARP inhibitors indefinitely. And one thing we really don't understand is, does the length of time somebody is on a PARP inhibitor contribute to a risk of developing a pre-leukemic syndrome? This is a very rare and uncommon event that has been observed in women who have been on PARP inhibitors. It is something we know seems to be a little bit more frequent when somebody's been on a PARP inhibitor than if they've never seen a PARP inhibitor. And there is some thought that the longer somebody is on a PARP inhibitor, potentially the higher the risk of getting this event called MDS, myelodysplastic syndrome, or AML, so leukemia, can be. And so I think that for me, certainly in newly diagnosed ovarian cancer, I do stop PARP inhibitors after 2 or 3 years, depending upon whether I have somebody on olaparib or on niraparib, based upon the clinical trial data. And in the setting where somebody's cancer has come back and I'm doing maintenance PARP inhibitors, it's a much longer discussion, because the data have really been based upon women continuing on PARP inhibitors indefinitely, but there is a risk there. And so it's something that we have to consider.
And you say it is a relatively low risk. About how high of a risk is getting one of those cancers? So,
in women with newly diagnosed cancer, we think that sort of that risk is probably somewhere in that 1 to 2% range.
Mm-hmm.
When we're looking at the trials that have been done in women with BRCA mutations and sort of, having PARP inhibitor indefinitely, that risk does seem higher. The SOLO-2 study reported a 8% risk of an 8% recurrence of MDS and AML. That is the highest that has been reported in any study. So I think that that's probably an outlier. But I think it may be up into the 2% range.
Okay. So still, it's a risk, but a low risk.
It's a low risk, but it is something we definitely have to think about. Yes.
Okay. Thank you for that. And so we have gotten some questions about rarer forms of ovarian cancer, so clear cell in particular, and low-grade serous. So I was wondering if you could speak about whether there's any, new research or treatment updates on that. And then I will just, before you answer, just give one plug that we did have a rare ovarian cancers webinar specifically in March, if anyone wants to view that or has more questions, and that's available on our website.
Okay. Great. So I think, rare subtypes of ovarian cancer are absolutely a research focus. I didn't present anything today because there wasn't really sort of any new information that came from SGO on those, but there's been a lot of work in the rare ovarian subtypes. Low-grade serous has had a lot of research being done recently. Some of you may know, based upon studies that have come out over the past 2 years, that MEK inhibitors are a new type of targeted therapy that we absolutely think about for low-grade serous ovarian cancers. and have shown activity that is at least as comparable, and one study has looked better than both chemotherapy and hormonal therapy in low-grade serous ovarian cancers. There are other types of therapy that are under development. So there are combinatorial strategies with MEK inhibitors that people are doing research on in low-grade serous ovarian cancers, as well as ovarian cancers that have KRAS mutations. And then there is a really interesting phase 2 trial that is being run through the GCIG groups, so with ENGOT and GOG Foundation. So these are cooperative group trials, not cooperative group, I'm sorry, these are sort of the larger sort of group trials, because these are rare tumors, but this is an international trial that's looking at the combination of a drug that both inhibits MEK and RAF. So those are both components of RAS signaling, together with a FAK, another sort of protein called FAK inhibitors. And they had a phase 1 trial that actually showed that this particular combination could be active even in women who with a low-grade serous ovarian cancer that have previously progressed on a MEK inhibitor. So that's an ongoing trial, very exciting. There's also an ongoing trial that's examining the role of hormonal therapy versus chemotherapy in newly diagnosed low-grade serous ovarian cancer, and whether or not we really need chemotherapy in that setting. So that's low-grade serous ovarian cancer. Clear cell carcinomas of the ovary have been a little bit, or are a harder nut to crack. These are very challenging cancers. There have been a number of mutations that have been identified in clear cell ovarian cancers, including something called ARID1A. And there are trials that are in development that are really looking to try and target ARID1A through a number of mechanisms, including using potentially ATR inhibitors, and what are called bromodomain inhibitors. There's also been a lot of sort of thought about whether immunotherapy might be particularly active in clear cell carcinomas of the ovary, and that came out of the first immunotherapy paper in ovarian cancer with nivolumab. That was only 20 patients and came out of Japan, but both clear cell patients who were on that trial actually had a response. And so there has been work looking at clear cell ovarian combination, immunotherapy combination strategies. Dr. Dizan down at Rhode Island has a combination of nivolumab and ipilimumab in clear cell carcinomas of the ovary that's been enrolling.
Great, thank you for speaking to that, because I know a lot of it focuses on high-grade serous, but since we do get some of those questions about rarer types, I always like to include those as well. So I think we're starting to run short on time, so we may only have time for one more question. And so I know that you talked about that really exciting trial that's about the phase 3 is about to start opening for AVB 500, which I had no idea about. So that's really exciting to hear. But I'm just wondering if you could just generally speak, aside from that, what is the best hope for platinum-resistant, platinum-refractory patients, or what are the treatment recommendations for them currently?
So I think there's, in the platinum-resistant setting, we still come back to chemotherapy. That's our standard of care, and there are chemotherapies that have shown activity. I think there are definitely new drugs on the horizon. There are antibody drug conjugates, which are basically targeted chemotherapies. They are an antibody that's sort of linked to a really toxic chemotherapy, but brings it right to the cancer cell. So it allows us to administer that really toxic chemotherapy. And those are in development. There is one drug called mirvetuximab that's currently in a phase 3 trial called Mirasol, which we're very hopeful about, that will have activity in women with folate receptor alpha expressing ovarian cancers, including those who have platinum-resistant cancers. We have, I think, combinatorial strategies on the horizon, and there are a lot of combination strategies, I think, that are targeting the development of PARP inhibitor resistance. So somebody who's had a PARP inhibitor and whose cancer has now become resistant after getting a PARP inhibitor, sort of thinking about ways to resensitize the cancer to PARP inhibitors. And there's a lot of research on that on the horizon. I think For anyone, for any person with a recurrent ovarian cancer, absolutely. I think that beyond, standard of care therapies, absolutely think about clinical trials. They are opportunities and hopefully ways for us to move our field forward.
Great, thank you. So I think that's a good ending point. And so thank you so much for being here today, Dr. Liu. This was a really informative program, and I feel like we, covered a lot of information. And then thank you to everyone who joined us today and for submitting all of your great questions. I'm sorry we couldn't get to everything, but we tried to cover a wide range of topics. I do ask that you please take a moment to fill out the survey at the end of the webinar. We really rely on your feedback to continue to improve our programs and make sure that we're offering topics that are of interest to you. So the survey is going to show up in the browser when the webinar ends, and then a link to the survey will also be included in the follow-up email that's going to go out. So you can take it either one of those ways. So thank you again, everyone, for attending. And thank you so much, Dr. Liu, for your great presentation and answering all of these questions.
Thank you so much. Appreciate it. Have a good day.
You too.